I'm Keiko Hirayama, a Wolfram Alpha developer in Scientific Content Group. I will be talking about neuroscience in the Wolfram language. It is our ongoing effort to add more data contents in medicine and life science area. And neuroscience is one of the subject area we just wanted to cover as there are growing interest among students in brain and cognition. I'm Keiko Hirayama, a Wolfram Alpha developer in Scientific Content Group. There are many ways in Mathematica to work with neuroscience data, ranging from processing images and signals to analyzing temporal data and network graphs. In this talk, we are specifically looking at data that had been added to Wolfram knowledge base to demonstrate how we could analyze and illustrate the structure and function of the human nervous system, all in the Wolfram language. And also it's part of our ongoing effort to expand this knowledge base in life science and medicine areas, as there are growing interest, especially among students in those subject areas. So with a newly introduced set of data, we can access information on brain anatomy, neuron characteristics, and cognitive map. And first, two set of data describe the structure of human nervous system, and the last one illustrates the functional aspect of the human nervous system. So starting off by brain anatomy, the brain is the central part of the nervous system, and the physical map atlas of the brain and network connectivity give us information about how our brain is wired in the nervous system. And our human brain contains about 100 billion of neurons, and of course, it's not an easy job to identify all of those brain cells. Characteristics of a few hundreds of neurons were identified already, for instance, physical and spiking characteristics, and those define the processing ability of the network. And thanks to the recent research with fMRI, we can detect localized brain activity when we process specific cognitive function, and that allows us to create a statistical cognitive map of brain activities. So we are looking at some examples for each of these. So starting off by brain anatomy, we have expanded existing anatomy data to include more detailed brain's regional anatomy, brain anatomy information. And our data is based on UMLS and NeuroLex. Both are lexicon type database, and the last one, Body Parts 3D, provides our graphics 3D models. And just to remind you again, all of the data and examples I'm going to show you is available through entity framework. So we have over 3,000 entities to describe brain structures, ranging from large cortical areas to smaller nucleus that constitutes the larger parts of the brain. And we have now over 120 properties to describe or access more information about individual structures. Some are image properties, graphics, and geometric properties, and some describes the connectivity or physical characteristics. And among those, some properties are especially dedicated to explore neuroanatomy. And with combinations of these entities and properties, we can take a more closer look at individual brain structures. For example, by accessing image properties of anatomy data, we can visualize the structure viewed from different perspectives. So this is looking at the large cortical area, prefrontal cortex. Or we can label and also color each subpart of the structure using anatomy plot 3D function. And of course, we can also ask Wolfram alpha to get more information. For example, what's the function of the prefrontal cortex, and get the normalized anatomical functional concept, decision making in this case. And there are many ways to separate brain areas in smaller regions based on structural characteristics. And Brodmann area is one of the classical categorizations to separate brain areas based on site architecture. And we can highlight the area of your interest or use additional properties to get more information. For example, we can get neighboring structures or find what larger systemic brain part this structure is part of. And moving to even smaller structure, for example, we can find the area, one of the areas of the brain associated with that encodes emotional state. And one is amygdala, which is the primal part of the brain and has key roles in emotional response, memory and recognition associated with memory, emotion, sorry. So once again, we can visualize the structure using anatomy plot 3D function. And amygdala is located near the center of the brain, and shape and size is just like an almond. And taking this amygdala as an example, we can now take a look at brain's wiring. We have neuronal connectivity information, which includes more than 1,500 input-output connectivity relations across different brain areas. For example, we can find output areas of amygdala there, and we can also visualize the network using the graph function. And this is the amygdala, and these are the direct output areas. And additional graph functionality in Mathematica help us analyze the network and find loop. Finding feedback loop is one example, which is the key framework in any biological network to fine-tune the system by altering the signal transmission. So for example, we can highlight the loops involves amygdala and other areas. For example, thalamic nucleus. Or next example, we can find specific circuits that involves particular structures. Here we are finding a circuit that involves amygdala and prefrontal cortex, and highlight in the graph. And as you remember, prefrontal cortex functions in decision making. So this circuit is crucial for modulating response to emotional experiences. And of course, we can find more than one circuits or loops in the network, but just for the demonstration purpose, I only show the 2 layers of the network. Also, the signals from the brain eventually reach the spinal cord to excite motor response, and sometimes network needs shortcut pathway to execute the command as quickly as possible. For example, to escape from the threatening object. And so minimum cost flow might be one of the ways to find those shortcut pathways. For example, amygdala to spinal cord. And not limited to brain's central network, we have also information about more peripheral nerve pathways. This is the example of visual perception pathway, and we can visualize them. So the light signal comes into the eye, and signal travels optic nerve crossing optic chiasm and reaches the visual area in the occipital lobe. And the brain integrates the input with other information and sends command back to periphery for motor response. So for example, we can find nerves innervating in your left hand and visualize those nerves. So now let's take a look at the even smaller components of the brain. Brain's functional unit, a neuron. We have information on over 300 types of neurons based on NeuroLex and the NeuroElectro database. And this is the morphology of a typical neuron. This is the soma of a neuron that contains cell body, and these branches are the dendrites, that's where the signals come in, so input areas. And the signals travel down the axon and reach the axon terminals, and where it makes synaptic connections with other neurons, so it passes the signal to other areas of the brain. And we have 33 properties to describe physical and spatial characteristics of different types of neurons. And some are about axons' specific properties, for example, origin of axon length or soma diameter, or some are about dendrite, number of dendrites, and dendrite location, and so on. And one of the important characteristics of the neuron is type of chemicals released from the cell called neurotransmitter. For example, we can find neurotransmitter released from one of the Purkinje cell of the cerebellum. And if you like to know more about this chemical, you can also access chemical data to get more information. And other types of properties describe the location of the cell, or reversely, you can ask what type of neurons are found in a given anatomical structure. So retina contains various types of neurons, including light-sensitive photoreceptor cells. Or even more specifically, we can find what type of neurons extends axon in particular area, for example, auditory cortex. And neurons use electrical signals to communicate with other neurons. And the researchers typically record the electrical spike activity called action potentials to identify the type and structure of the signal transmission. And of course, they can use our signal processing functionality to sort out spikes and analyze their data. And we have some characteristics, properties to describe those electrophysiological characteristics of those spiking activities or action potentials. Some important characteristics are amplitude, width, or threshold of the spikes. And we can take a look at spike frequency of various types of neurons. And we can see how some neurons are fast firing versus some are slow in firing. And these variations define the processing ability of the network. And this is just a small excursion from the main topic, but we can also model spike neurons. Mathematica is fully equipped with solving differential equations, so we can take advantage of that and model spike activities using Hodgkin-Huxley model. There are a number of variations in Hodgkin-Huxley models, and some allow us to simulate the propagation of spike activities from population of neurons. And we can easily change the parameters to find the best fit model for the biologically biologically plausible spiking neurons. So finally, after looking at the smallest components of the brain, let's take a look at brain's cognitive function. A variety of cognitive processes that takes place in the brain can be categorized and hierarchically mapped. So we have over 800 concepts from Cognitive Atlas database to describe brain's cognitive function, and some properties are available to get more information about individual concepts. For example, definition, associated anatomical site, and some hierarchical relationships. And by looking at these hierarchical relationships, we can illustrate how some of our fundamental cognitive functions, for example, decision-making, memory, and perception are related to one another. For example, here, memory, perception, and decision-making are highlighted here, and it's connected. For example, perception, especially auditory perception, and here memory recognition meet here for tone recognition, or here face recognition, and from decision making, social inference meet here for facial trustworthiness recognition. And sometimes other type of visualization help us gain some more insights. And this is looking at the same relations, but with using community graph plot. Group of concepts here describe the perception, and here constitute the memory, and this constitutes the decision making. So they all meet here in the middle for recognition of object, face, and patterns. But we can also relate to this to structural parts of the brain. For example, we can find cognitive concepts associated with each cerebral lobe of the brain, and find a simple, create a simple map like this. For example, frontal lobe functions in motor control, decision making, and so on. But this is just abstract representation of the brain function. But now we can take a look at more detailed brain activities. We have fMRI is powerful tool to detect subtle change in brain activities. And we have statistical maps of brain activity from database called Neuroboard, and which is based on meta-analysis of task-based fMRI. So this is the original data. It's a voxel data. And we converted this to graphics and also aligned to the coordinate system used in anatomy data. For example, this is looking at brain images, coronary sliced from front to the back during decision-making processing. So we can spot the high activity area near the top surface of the brain. So how did they measure this? There are standard experimental tasks to assess those certain cognitive abilities. For example, these are the tasks to measure decision-making ability. And if you are not familiar with those tasks, we have also basic information about each task. And of course, you can use our interface functionality to build your own task set just for some fun. This is our gambling task to measure decision making, or image recognition test to assess visual object recognition ability. But these are just some examples to assess cognitive ability. So more examples of brain activity. We have arithmetic processing, our math or calculation ability, involves parietal lobe of the brain, indicated by these high activity areas. And next one is language processing. We can also make an image collage with just a simple line of code. This is looking at the brain from the front, and this is coronal slice, horizontal slice from top to bottom, and sagittal slice is from right to left. And in most people, language area is located in the left side of the brain. And next one is emotion processing. We can spot the higher activity area near the center of the brain. Perhaps this shows your memory. This is amygdala. And this activity area is available in the form of 3D graphics and can be extracted. So it's a rotatable activation area associated with emotion. So we can combine this graphic with other brain structure models in anatomy data to, for example, get the graphics overlay or do some geometric computations to actually calculate the graphics intersections. So finally, this graphics combines the hierarchical organization of the cognitive concept with brain activities. And this top is the visual perception, and we are looking at child concepts. And it's interesting to see how our brain categorizes visually perceived place, pattern, or number, string, with all distinctive patterns of activities. So now I'd like to take a moment to summarize my talk. With a newly introduced set of data available in Wolfram language, provide us information about new brain's anatomy, and neuron characteristics, and statistical cognitive maps. And neuroanatomical divided areas describe the structure and function of the nervous system. And the connectivities across those brain areas can be characterized and visualized. And of course, additional graph functionality in Mathematica help us analyze the network. And also we have information about individual types of neurons, and those can be characterized for spike analysis. And finally, we also have statistical maps of brain activity to illustrate or visualize the brain activity associated with cognitive function, and those can be further processed or extracted for the graphics computation. So what's coming next? We are expecting to add more information about individual brain circuits and sensory motor pathways. And as you might heard about it, there are ongoing human connectome map project where they are looking at connectivities in fiber levels. So there are more to be discovered and data to be expanded in the near future. Also, we welcome any suggestions or requests for data addition in computational neuroscience area. So that's all I have now. Thank you very much. Yes.
Yeah, quick question. Do you have anything related to pathology?
Yeah. So anatomy data has associated disease cause, like properties, to describe what disease are associated with individual anatomical structures. But we don't have anything about more detailed pathology and so on. Just what disease are associated.
With a certain area?
Yeah, certain area of the brain or certain structures in the body, and not limited to brain anatomy. Yes.
Everything except the last slide is included in 11.2?
Yes. Yeah, thank you. Yes.
Related to the Connectome project.
Yeah.
Is there any plans to introduce clarity images as well?
Which image, sorry?
Clarity with I know it's postmortem, so hopefully not on humans, but maybe
Yeah. Yeah. Actually, we are looking at other species. Actually, we have some plans of adding. Yeah, I have actually another slide. So, yeah, this is another project. We have animal anatomy project. So we have plans of adding some structures in different species.
That's more like a side project.
Yeah, this is side project. Yeah. Yes.
Are you familiar with something called Brian?
Brian, yes, I heard about it.
Is it possible to use similar simulations?
Yeah, I think so. We have like equipped with all interface functionality, so it's I think possible, but it's just not in our entity framework. But I think it's possible.
Another question. Do you have extension to the peripheral nervous system?
Yes, we have models of peripheral nervous system. So, but of course, it's not complete. Some of smaller nerves are still missing, but we have a large number of peripheral nerves. So, for example, So this one is looking at large major nerves innervating the hand or arm.
So could you use it to interrogate the system about someone comes in and they can't move their right arm,
Yeah.
let's say. Could you interrogate the system about where the possible lesions?
Yeah, that's something we don't have those data yet, but I think that's something.
I mean, you may not need any data at all. I mean, if you have.
Okay, so we can just, but yeah.
You can start adding a medical disclaimer to mathematical.
Yeah.
We are not doctors, we only play them on stuff.
Yes.
Also, you have the major modes of variation with all these anatomical structures, like, people are come in different shapes and sizes, so if I want to
Yeah.
compare this to actual measurements, I may have to have those variation models.
Yeah, that's right. Yeah, it's only like, for example, this is male adult models. Of course, there are variations in males versus females and also children versus adults. So that's something we have to address. Yeah.
That looks already like an amazing amount of data
Yeah.
hooked up and interconnected and put in that.
Yeah.
Many of the quantitative data about bones, whatever, have ranges for sizes. So it's not just the model we have.
Well, usually you just measure a whole set of bones, and then you just find out just what are the most linear modes of variation. And if you just take the first 5 and so on, that's a pretty good idea what variations can occur.
Not sure what exactly you mean by variation.
Well,
Do
just
you guys want
you
more
take-
questions?
Okay.
We can get ready for the next presentation.
Thank you.
We can discuss more.